Good morning, students. Today we will study the internal structure of leaf. First, A part is the rachis. Rachis is in transverse section of rachis. It is a circular in outline. It has two rows of leaflet inserted on the adaxial side. The rachis is differentiated into epidermis, hypodermis, ground tissue, and vascular tissue. The outermost layer forms the epidermis. It is covered with a thick cuticle. The epidermis is interrupted by sunken stomata. Each stroma consists of two guard cell and two subsidiary cell. The epidermis is followed by hypodermis, which is composed of two types of cell. The outer 2 to 3 layers are composed of thin wall chlorenchymatous cell and the next 4 to 5 of sclerenchymatous cell. There are 2 to 3 layers of sclerenchymatous cell on the adaxial surface of the rachis. Next to the hypodermis is the parenchymatous ground tissue with many mucilaginous canal. There are several vascular bundles in the ground tissue and they are arranged in a typical inverted omega-like arc. The vascular bundles are conjoined, collateral and open, and each is surrounded by single layer bundle sheath composed of thick wall cell. They are diploxylic, that is, they have both the centrifugal and centripetal xylem. The arrangement of xylem and phloem differ in vascular bundle in the basal, middle and the upper region of the rachis. The vascular bundle in the basal region have only centrifugal xylem. The protoxylem in this bundle lies toward the center of the rachis, that is, they are endark in nature. A little higher up, the centrifugal xylem is reduced, only a few protoxylem elements remain endark and the rest begin to move in a lateral direction. In the middle of the rachis, group of thick wall cell develop just behind the protoxylem elements and these finally differentiated into centripetal xylem. The vascular bundle in the upper region of the rachis have more centripetal xylem than the centrifugal xylem. At this stage, centrifugal xylem lie in a small group on both the side of the centripetal xylem. At the extreme tip of the rachis, centrifugal xylem may be completely absent. It is generally believed that centrifugal and centripetal xylem have a different origin. Centripetal xylem start differentiating at a very early stage of development and its lignification occurs slowly, but centrifugal xylem originate by secondary growth. The B portion of the leaf is the leaflet. There is a single midrib in the leaflet. Lateral veins are absent and the leaflet is swollen in the middle region. The margin of the wings are revoluted in case of Cycas revolata and straight in Cycas circinalis. The outermost layer form one cell thick epidermis which is covered by a thick layer of cuticle. The upper epidermis is continuous whereas the lower epidermis is interrupted by sunken haplostellic stomata which are confined to the wing. The epidermis is followed by a layer of sclerenchymatous hypodermis on hypodermis. Hypodermis form a continuous layer on the dorsal side, but on the ventral side it is confined only to the midrib region. In Cycas revolata, however, hypodermis is also present in the wing. The hypodermis serve as a screen and protect the plant from overheating and excessive transpiration. Next to hypodermis, there is a well-developed mesophyll tissue which is differentiated into palisade and spongy parenchyma. The palisade is a single layer, continuous layer of columnar cell. The spongy parenchyma consists of several layers of loosely arranged oval or irregular cells that are confined to the wing. The cell of both the palisade and spongy parenchyma are rich in chloroplasts. There are group of trachid-like cell on the lateral side of the vascular xylem in the midrib region. These cells are small and broad and have reticulate thickening or broader bordered pit on their wall. They represent the transfusion tissues. In the wing region, long, colorless, transversely elongated parenchyma cells are present in between xylem, in between the palisade and the spongy parenchyma. They represent the secondary or the accessory transfusion tissue. The secondary transfusion tissues is connected with the centripetal xylem, affecting the on the bundle side. Since lateral veins are lacking in Cycas, accessory transfusion tissue help in the lateral conduction of water and nutrient. A single vascular bundle is present in the midrib region. It is surrounded by parenchymatous bundle sheath. The vascular bundle is conjoined, collateral, open and diploxylic. It has a large triangular centripetal xylem and two small group of centrifugal xylem, one on either side of centripetal protoxylem. Phloem is present on the abaxial side of the xylem. It is composed of parenchyma and sieve tube. A thin layer of cambium is present between the xylem and phloem. Thank you.